Datta biology classes. Blood is a liquid connective tissue. Blood is a liquid connective tissue. So blood is a liquid, it is a liquid in nature. Here this is blood, this is liquid. Connective tissue because it connects the different cells of an organisms, different living cells. Everywhere, anywhere you go into living cells, you will get blood. So blood connects each and every cells or each and every organ, systems and everything. Why it is tissue? Because it contains various cells that is erythrocytes, leukocytes and thrombocytes, but they perform the main function. They perform a uniform function of circulation. The main function they are doing is circulation. Main function. Now, why the blood remains liquid? Because blood contains two things, solid part and liquid part. Solid part and liquid parts. The solid part is formed of three. One is erythrocytes, then leukocytes, and thrombocytes. These three formed elements, they are also called formed elements. And liquid part is known as plasma. P-L-A-S-M-A. Plasma is the liquid part. Now, this formed element remains suspended within the blood. Erythrocytes, so many erythrocytes remain. Leukocytes and thrombocytes. These are cells or formed elements remains suspended within the blood, that is plasma. Within the plasma it remains. The plasma portion is 55% and solid part is 45%. That is, out of 100, 55 remains liquid. So it remains in a liquid state. And solid part is less, so it remains solid. It does not coagulate because inside this plasma there are some mechanism by which anticoagulant factors do not allow the blood to clot. Okay? Now, how this 45 and 55%, these are called hematocrit value. This 40-55 is to 45 is known as hematocrit value. How this is ascertained? How this is ascertained? How this is ascertained? Take a test tube. within this test tube you put some blood. Put some blood. The test tube should be graduated, millimeter, centimeter wise it is graduated. This is called Winthrop's tube and you are putting blood inside. So this blood will contain erythrocytes, leukocytes and thrombocytes, and there will be liquid part also. So once you are putting it, it will coagulate, it will clump. In order to avoid coagulation, you have to put something. You have to put some anticoagulant. Okay? This anticoagulant are say ACD, acid citrate dextrose, and sometimes the laboratory people use EDTA, or in hospital also they are used. Ethylene diamide tetraacetic acid. Ethylene diamide tetraacetic acid. These are the anticoagulants which is put inside this tube to make the blood, to keep the blood in a liquid state, so that it does not agglutinate or it does not coagulate imperfectly. Then this solution is closed here. You tighten the cap and put this in a machine, centrifugal machine. That centrifugal machine revolves 3000 times per minute. It should be continued for say 30 minutes. How much? This blood is to be put in a machine, centrifugal machine, and they will be, it revolves 3000 times per minute and it should be continued for 30 minutes. Even then blood will not coagulate because anticoagulant I have added. And you can say whenever it is revolving, these cells, these small elements are so delicate, whenever it touches the wall of the tube, it may disintegrate, it may burst. So it should be provided with some wax, or should be made as such that even if it handles like airbags in a car, even if it collides, but it will not burst. So we will see that these 45% gets deposited here or sedimented here, and these portions remain clear liquid. This is called 55%. This is plasma. 45% and 55%. This is called plasma. So in this way, hematocrit value is ascertained. Now, this is just hematocrit value. This is why it remains in liquid state. Okay. Our main intention is what is there inside the blood. Erythrocytes, leukocytes and thrombocytes. I have erased it, thrombocytes. Thrombocytes. If the erythrocyte increases within the blood, a condition is known as erythrocytosis. And if it decreases, it is known as erythrocytopenia. Cytopenia. That is, it decreases, it increases. Erythrocytosis, erythropenia. Clear? Leukocyte, if it increase, called leukocytosis. If it decreases, it's called leukopenia. P-E-N-I-A, leukopenia. If thrombocytes increases, it's called thrombocytosis. If it decreases, known as thrombocytopenia. Quite simple. Erythrocytosis, erythropenia; leukocytosis, leukopenia; thrombocytosis, thrombocytopenia. As simple as that. Let us make it a little bit complex. If this increases, this is 2, this is 3. This is known as polycythemia. Polycythemia. P-O-L-Y, poly, C-Y-T-H-A-E-M-I-A, polycythemia. A disease known as polycythemia vera. This erythropenia is otherwise called, that is less RBC, it is called anemia. A-N-A-E-M-I-A, anemia. If leukocyte increases to a considerable extent, leukemia. Leukemia is blood cancer. Thrombocytosis have two forms. Thrombocytosis, if it increases, it is called thrombocytosis. And thrombocytopenia is less thrombocytosis, blood do not clot. If there is less of this, it is thrombocytopenia. It is sometimes called thrombocytopenic purpura, a disease where blood does not get clotted. Whenever there is a cut, the blood oozes out through the cut. So these are most thrombocytopenia. And thrombocytosis, you have come across two words, cerebral thrombosis. Then cardiac thrombosis. Cardiac thrombosis. Here the blood causes thrombus, that is, it coagulates within the minute vessels, of the cerebral region, that is brain, cerebral thrombosis, or within the heart. It creates thrombus, that is, it cannot pass on. So the heart remains devoid of nutrients or oxygen, the cell dies, and in case of cerebral, the cell dies. So there are two terms, cerebral thrombosis and cardiac thrombosis. Okay. Now our part of discussion will be on erythrocytes. This I am erasing out. Okay. Now, these erythrocytes, how much it remains? 1 millimeter, 1 millimeter, 1 millimeter, 1 millimeter. 1 millimeter, 1 millimeter, 1 millimeter, 1 millimeter. Within this 1 cubic millimeter, there are 5.4 million RBC, erythrocytes. Within this 1 cubic millimeter, 1 millimeter, 1 millimeter, 1 millimeter, 1 millimeter, everywhere it 5.4 billion for mil. See how much is the quantity. In case of female, it is 4.8 million for female. This is erythrocytes remains. Now, the variation, here I have said the number, increases the number or decreases the number causes anemia. This one. Anyway, I will come later on for anemia. If there is variation in size, variation in shape, variation in size. Say for example, variation is in size of the erythrocytes. Erythrocytes are like this, biconvex, biconcave. This is the structure. It's biconcave. Nucleus do not have nucleus. This and that, this and that. Now, if the size of this varies, some size of this, some size of this, some size is even more bigger. So variation in size. This variation in size is known as anisocytosis. Aniso, not similar. Iso means similar, aniso means not similar. So there are different shapes, different sizes. So one is small, one is middle, one is big. This and this. If this remains within the blood, say 3 or 4 or 10 lakhs like that. So it is the situation or condition is known as anisocytosis. Then variation in shape. Variation in shape. This is the shape. Variation in shape. Suppose some RBC like this, some RBC like this, some RBC like this. So these are different shapes. So in case of variation in shapes, it is known as poikilocytosis. POI, poikilo, KILO, poikilocytosis. Very easy. So what we have learned, erythrocytosis, erythrocytopenia or anemia, polycythemia or polycythemia vera is the name of the disease. It is the more than enough. It is little bit more, no problem. But it is further than if it increases, then it causes polycythemia, a disease. Anisocytosis different in size, poikilocytosis is known as different in shape of the RBC. Now, the production process, how it is produced. Production process of RBC. The production process of RBC is called erythropoiesis. Erythropoiesis is the name given to the formation of RBC. The RBCs are formed through a process called erythropoiesis. Now, how it is formed? There are two theories advanced for this erythropoiesis. One is given by British, that is Europe, and USA has given. Two theories they have advanced, almost same, but they have given two theories. First theory, they have called the initial erythroblast. They are calling it megaloblast. They are calling it megaloblast and they are calling it erythroblast. It where does RBC forms? It forms within the bone marrow. Where is bone marrow? Within the bone. So if you write a bone like this, in these two sides, these are called epiphyses. In this area, in the red bone marrow, in the adult, the RBCs are formed. RBC are of two types, that is hemoglobin. Anyway, these RBCs are formed within the bone marrow. British people are calling it erythroblast, and these people are calling it megaloblast. After some times they have come to an agreement. This erythroblast, they are writing it erythroblast. Erythroblast. They are saying is early erythroblast, late erythroblast, and mature erythroblast, like this erythroblast. Then they are coming, both of they comes and say normoblast. Normoblast. These are not that much important, but these are the procedure or the production process. Normoblast. They have come to a conclusion. Yes, it is normoblast. Normoblast. After the formation of normoblast within the bone marrow, it becomes reticulocyte. This point is to be remembered. Reticulocytes. They are also calling it reticulocytes. So both are same. They have forwarded this theory, they have forwarded this theory, because we, the people to whom we follow, we do not follow anybody, we follow both of them. So these are the process they have given. From reticulocytes, they are giving the name of erythrocytes. Then these erythrocytes. Erythrocytes, CYTES, erythrocytes. These are also erythrocytes. So erythrocytes are formed. Erythrocytes are formed. Now the point to be remembered, I told you that up to the formation of reticulocyte, the nucleus remains. Here the nucleus remains within the cell, nucleus remains in both the cases. But once reticulocyte is produced, the nucleus vanishes. That's why I call that mature erythrocyte do not contain nucleus other than camel invertebrates. Okay? So how this erythropoiesis takes place? Now I will come to that. How this erythropoiesis takes place within our body. Who advises them to produce erythrocytes? Erythrocyte, production of erythrocyte is known as erythropoiesis. How it is happened? We have one pituitary gland in our cerebral hemisphere in the brain. We have some pituitary gland. This is anterior pituitary, this is posterior pituitary. Pituitary gland, this is the master gland. This pituitary gland produces a hormone called adrenocorticotropic hormone, which we will discuss during the chapter of hormones. This is adrenocorticotropic hormone. It is a tropic hormone. What is the, why it is then given as tropic hormone? Because where it is produced, it does not act there. It acts on a distant area. This is adrenal cortex. Adrenal cortex. In adrenal cortex, it stimulates the adrenal cortex or gives intimation to adrenal cortex to release erythropoietin releasing factor. They produces erythropoietic releasing RELEASING factor. They give information to bone marrow. To whom they give? This bone marrow. bone marrow then produces or starts active to produces from megaloblast, normoblast, armiblast, blast, then erythrocytes. These are the process. So this production is known as erythropoiesis. Okay. Now we'll come to the next part, or rather penultimate part of our discussion. This erythropoiesis. Then we'll come to another point, that is production. What is the rate of production? Erythropoiesis, like in this way, at the rate. Erythropoiesis at the rate 1.5 million to 2 million per second. Within a second, 1.5 to 2 million produced. Another second, 2 billion produced. Another second, 2 billion produced. Astonishing rate, the erythrocytes are produced. So no problem. If anybody hit you or cut you, don't get afraid, because you are getting 2 millions for 1 second. No need of having panic. Okay. So this is 1.5 to 2 million are produced per second, and this is in cubic millimeter of a blood. That is, this is the blood, total blood. If you take 1 cubic millimeter of this blood, this cubic millimeter contains 5.4 to or 4.8 million of erythrocytes within the cell. So no need of panic or getting afraid of this. Now I will come to, this is called anemia. There are various types of anemia, which I will come one by one. This is very simple, not difficult. I am erasing all this thing, because this, if you go through, just write this in this way so as to remember in a nutshell. Anemia. First, what is anemia? That is RBC, relates to some RBC. Either there is no RBC, either there is defective RBC, either there is the, so many things are there. First, I will talk about aplastic anemia. Number one is this one. Aplastic anemia. A-N-A-E-M-I-A. Aplastic anemia is a very dangerous disease, because it, in this case, what you know, the cells of during erythropoiesis within the bone marrow, something happens within the bone marrow, megaloblast, early erythroblast, normoblast, reticulocytes, this production system stops. That is, it destroys the bone marrow cells. You write bone marrow cells are destroyed. So there is no production of RBC. Production unit is closed because of some problem. Production unit is closed. So bone marrow destroyed, aplastic anemia occurs. It may lead to fatal. If you go on giving blood transfusion, how much? How long you will give? So it is a very serious condition, life-threatening. Next one I will write. It will become in red. Hemolytic anemia. H-A-E-M-O, hemo, L-Y-T-I-C, hemolytic anemia. Number 2, hemolytic anemia. Hemolytic, that is something get lysed, that is RBC breakdown. You write breakdown of RBC. Is called breakdown of RBC is known as hemolysis, which causes hemolytic anemia. Breakdown of RBC, you know, within the blood, a tissue, within the blood tissue, here the RBCs are there. The RBCs are there. So within the RBCs, hemoglobin is there. So once RBC bursts, the hemoglobin comes within the plasma and it appears red. It is known as hemolysis. HAEMO, hemo, LYSIS. So in this anemia, breakdown of RBC takes place and there is increase in hemoglobin within the plasma. There is plasma comes within the hemoglobin comes within the plasma, and that is why this is known as anemia. Blood supplementation is necessary. Number 3, the number 3, thalassemia. THALA, thalassemia. Thalassemia. It is also a dangerous. So according to a dangerous situation, I am writing that thalassemia is a disease where it is inherited blood disorder of hemoglobin. You write inherited blood disorder. How? In order to produce hemoglobin within the RBC, that hemoglobin is formed by DNA. So within the DNA, whenever we will come to DNA, you will see ATGC, A, T, G, C, like this. A, T, then G, C, or G, A, C, T. In this way, amino nucleotides are arranged in double bond or triple bond, which I will come. So there are 3. There is a codon. This is called codon. This is one codon. This is another one. This codon is responsible for the formation of HB. That is hemoglobin. This hemoglobin, this codon gets mutated. Suppose it should be CGT. It gets mutated to CAT. So it gets mutated. This mutation prevent the production of hemoglobin. So it is an inherited blood disorder due to what should I write? DHB hemoglobin non-production, or what should I say? Mutation of genes. Due to mutation of genes for producing hemoglobin. This is known as thalassemia. There are varieties type. We will come during that chapter. Then we will write another thing: sickle cell anemia. Sickle cell anemia. Sickle cell anemia. This is also inherited disorder. Inherited blood disorder. Here hemoglobin is the action. They are inherited blood disorder where RBC gets sickle shaped, S-I-C-K-L-E, sickle shaped, S-H-A-P-E-D. If you write in this way, it becomes like this also. This is sickle shaped anemia. It is prevalent in African countries. It is also a gene disorder. Then this is pernicious anemia. Pernicious, pernicious anemia. Or it is called additions, pernicious anemia. Or it is called megaloblastic anemia. These are all same. Actually, in fact, it is body cannot produce vitamin B12, or rather body cannot absorb. You write body cannot absorb, absorb vitamin B12. Why? Vitamin B12 is required for erythropoiesis. This vitamin B12, if it does not get absorbed from the duodenum of our body, then the vitamin B, in absence of vitamin B12, erythropoiesis does not occur. So it causes this pernicious anemia or additions pernicious anemia or megaloblastic anemia. That is deficiency of vitamin B12. If you give vitamin B12 more by injection or by any means, then the disease can be cured. There is another one. This is also nutritional. This is called folate. Folate deficiency. Folate is actually vitamin B9. Vitamin B9 is also required for erythropoiesis. So there is less folate, less folic acid. In the body causes folate deficiency. Doctor used to prescribe some capsules with folic acid, vitamin B12, vitamin B6, so many vitamins along with folic acid they used to give, so it can be cured. Then another one is iron deficiency anemia. IROM, iron deficiency anemia. Now what does this iron do for causing anemia? In hemoglobin structure you will find Fe. Fe. Within the hemoglobin there is iron. It is an iron containing protein. It remains within the RBC, the hemoglobin. So iron deficiency is due to deficiency of iron. Deficiency of iron leads to deficiency of production of hemoglobin. Consequently, the iron deficiency anemia IDA occurs. Clear? It is getting more and more complex. One another term is there, normocytic normochromic. you have come across some blood reports. Their laboratory technicians used to write normocytic. What is hemoglobin? How much hemoglobin? They are writing normocytic, normoplastic. That is to give away with all these things. Fewer RBC than normal. Here you write fewer RBC than THAM than normal. What is the normal? Normal is 5.4 million, 4.8 million for female, and for male it is this. So if it is less than this, they used to write normocytic, normochromic, a little bit normal. You give vitamin or you whatever you have choice, you give some tonic phonic, so the patient may get recovered. So up to this, the erythrocytes chapter is over. We will go into the WBC in the next class. Thank you very much. Till then, goodbye.